Hello, friend. Welcome to Science for Sleep. Whether you have returned tonight or are discovering this channel for the first time, this is a quiet place to slow down, learn something gentle, and allow the day to gradually fall away. Tonight, we are beginning with a strawberry. Picture one resting in the palm of your hand. Its broad shoulders sit beneath a small green cap. Its body narrows toward a rounded point. The surface is bright red and slightly glossy, with shallow dimples holding hundreds of tiny yellow or brown dots. Cut through the middle, and the flesh is pale near the center, redder near the outside, moist, soft, and filled with a scent that can spread through a room within seconds. Few fruits are easier to recognize. Even before tasting one, most people can identify a strawberry by its shape, green leaves, and sweet fragrance. Yet nearly every visible part of it creates a botanical puzzle. The strawberry is called a berry in kitchens, markets, recipes, and ordinary conversation. In botany, however, it is not considered a true berry. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. The scientific meaning of berry has little to do with whether a fruit is small, sweet, round, or easy to place in a bowl. Botanists classify fruits mainly by the flower structures from which they develop. A true botanical berry generally forms from one flower ovary. The ovary is the part of the flower that contains young ovules, which can later become seeds after fertilization. Blueberries fit this botanical pattern. So do grapes and tomatoes. Even bananas, although they look nothing like garden berries, are classed as botanical berries. A strawberry follows a different plan. Its scientific name is Fragaria cross ananassa, with the cross marking its origin as a hybrid. It belongs to Rosaceae, the rose family, alongside roses, apples, pears, raspberries, cherries, peaches, and almonds. Its formal botanical category is an aggregate accessory fruit. The phrase sounds more difficult than the idea behind it. Aggregate means that many small fruit structures develop from one flower. Accessory means that much of the soft edible tissue comes from a flower part other than the ovaries. In a strawberry, the red flesh develops mainly from the receptacle. The receptacle is the thickened base of the flower, the small platform that once supported its petals, pollen-bearing parts, and many tiny ovaries. As the strawberry develops, this base swells until it becomes nearly the entire red body we recognize. The objects commonly described as seeds are stranger still. Each small dot on the surface is actually a separate dry fruit. Inside each one is the true seed. This means that one strawberry is not simply one fruit carrying many seeds within its flesh. It is a collection of many small fruits resting across one enlarged flower base. A medium-sized strawberry may carry roughly 200 of these surface fruits, though the number changes with the variety, flower size, and completeness of pollination. That unusual construction is only the beginning. To explain how the receptacle grows, pollen must be followed across a white flower, where developing seeds release chemical signals into nearby tissue. The modern garden strawberry also leads into a genome containing 8 complete sets of chromosomes and 2 wild American species that met in European gardens during the 18th century. Its red color leads into plant pigments. Its flavor leads into sugars, acids, and hundreds of airborne aroma compounds. Its softness leads into cell walls being carefully taken apart as the fruit ripens. Its brief life after harvest leads into fungal spores, rapid cooling, refrigerated trucks, and farms designed around a fruit that can bruise beneath a fingertip. All of that complexity is present in something that still looks simple enough to hold between two fingers. And the clearest place to begin is with one of the tiny structures scattered across its red surface. Choose one of the small dots resting in a shallow hollow on the strawberry's surface. Under a fingertip, it feels firmer and drier than the soft red tissue around it. Its outer layer has a faintly woody texture, and its color may range from pale yellow to deep brown as the fruit matures. This small structure is called an achene. An achene is a dry fruit that usually holds one seed. Botanists also describe it as indehiscent, which means that its wall stays closed at maturity instead of splitting open to release the seed. A sunflower seed is another familiar example of this arrangement. The shell-like part people crack is technically a dry fruit wall, while the true seed lies inside. On a strawberry, the same basic division exists at a much smaller scale. The visible outer surface belongs to the achene. A thin fruit wall surrounds a single true seed, where a young strawberry plant can begin if germination conditions are suitable. The seed contains an embryo, stored nutrients, and a protective coat. Its embryo already holds the first simple structures of a future root and shoot. When moisture enters and the temperature is suitable, the embryo can resume growth after a period of quiet dormancy. Across one strawberry, this arrangement is repeated many times. A medium fruit often carries roughly 200 achenes. Large cultivars may carry more, while smaller fruits or flowers with fewer functioning ovaries may carry fewer. The total depends on the cultivar, the size and shape of the original flower, how many ovaries formed, and how evenly pollination occurred. For that reason, a few hundred is more useful than treating 200 as a fixed count. Every achene began as a separate ovary within the same flower. Before the petals opened, those ovaries were packed closely across a small raised surface near the center. After fertilization, each ovary developed its own fruit wall and enclosed seed. The many achenes remained separated from one another, even as the tissue beneath them expanded and carried them outward. That tissue is the receptacle. In the flower, the receptacle began as a small green base supporting the petals, pollen-bearing anthers, and seed-producing structures. During fruit development, its cells divided and enlarged. Water entered those cells. Sugars arrived through the plant's transport tissues, and the receptacle gradually changed from firm green tissue into the soft body of the strawberry. The achenes remain attached to this enlarging base through tiny bundles of vascular tissue. Vascular tissue is the plant's internal transport system. One part carries water and dissolved minerals upward from the roots. Another moves sugars and other substances from the leaves into growing tissues. These narrow connections help support the developing achene and its seed, while also linking its development with the receptacle below. The surface of a ripe strawberry therefore preserves the layout of the flower that produced it. Each small fruit occupies the place where one ovary once rested. As the receptacle swelled, the spaces between those fruits widened. Their shallow pits became more visible because the softer surrounding tissue rose around them. At the broad upper end of the strawberry, another part of the flower remains in plain sight. The green cap is called the calyx. It is formed from leaf-like structures known as sepals. Before the flower opened, the sepals folded around the bud and helped protect its delicate inner parts. After the petals fell, the sepals remained attached, while the receptacle enlarged beneath them. They may stay green for days after harvest because their cells still contain chlorophyll, the pigment plants use to capture light. A strawberry can now be seen as a compact arrangement of many dry fruits, each carrying one seed, all supported by a single enlarged flower base. Its final shape depends on how successfully those separate ovaries begin developing across the entire flower. Before any achene forms, the strawberry begins as a small white flower held above the leaves on a slender stem. 5 rounded petals usually open around a yellow center. Near the outer edge of that center sit many pollen-producing anthers, each carried on a fine stalk called a filament. Together, an anther and its filament form a stamen, the male part of the flower. Closer to the middle rises the young receptacle, still small, firm, and green. Its surface is crowded with pistils. A pistil is one complete female flower structure. At the top lies a sticky surface called the stigma, where pollen must arrive. Beneath it is a short style, followed by an ovary containing one ovule. After successful fertilization, that ovule may become a seed, while the ovary around it develops into an achene. A single strawberry flower can carry hundreds of these pistils across its raised center. That arrangement makes pollination a distributed process. Pollen reaching one stigma can fertilize one ovary, yet many nearby stigmas still require pollen of their own. Most modern garden strawberries produce flowers with working male and female structures. This allows pollen from the flower's own anthers to reach its stigmas. Movement may happen when the flower shakes in a light breeze, when petals and anthers shift, or when insects brush across the center. Self-pollination can produce fruit, although it does not always spread pollen evenly over the full surface. Air currents contribute some movement as well. Pollen grains are small enough to shift over short distances, but a strawberry flower's low position and tightly packed pistils limit how thoroughly wind alone can cover it. Insect visitors often improve the result. Honeybees may land near the yellow center and circle while collecting pollen or nectar. Bumblebees, solitary bees, hoverflies, and several smaller flies also visit strawberry flowers. As an insect walks across the flower, grains cling to hairs on its legs and body. Turning movements bring those grains into contact with stigmas located on different sides and heights of the receptacle. Repeated visits can spread pollen more completely than a single landing. The benefit can be seen later in the berry's shape. When most pistils are fertilized, the receptacle usually expands more evenly. Areas with weak pollination may remain flatter or form grooves, especially when several nearby ovaries fail together. Cold weather can add difficulty. Low temperatures slow pollen tube growth, which is the process by which a pollen grain sends a narrow tube through the style toward the ovule. Frost can injure the center of an open flower, sometimes turning it dark and preventing normal fruit development. Warm weather speeds many stages of growth, provided the temperature remains within a range the flower can tolerate. Extreme heat may reduce pollen quality, shorten bloom life, and interfere with fertilization. The position of a flower within its cluster matters too. A strawberry plant usually opens its primary flower first. This blossom often sits at the center or top of the cluster and commonly produces the largest berry. Secondary flowers open later and usually form smaller fruit. Tertiary flowers appear after them and often produce the smallest berries because they begin with fewer pistils and compete for resources after the earlier fruit has already started growing. From open flower to ripe strawberry, development often takes about 28 to 35 days. Cool conditions may stretch that period. Steady warmth can shorten it, while cultivar, water supply, and sunlight also influence the pace. During those weeks, every successful ovary changes from a tiny part of the flower into a developing achene. The seed forming inside does more than preserve the next generation. It also helps produce chemical signals that tell the red tissue beneath it where and how strongly to grow. Within hours of fertilization, each successful ovary begins changing. Its outer wall starts forming the dry shell of an achene, while the ovule inside develops into a seed. A tiny embryo takes shape within that seed, carrying the first cells of a future root and shoot. At the same time, the young achene begins influencing the much larger receptacle beneath it. Its role extends far beyond protecting the seed. Developing achenes become important sources of auxin, a family of plant chemicals that regulate growth. One common natural auxin is called indole 3-acetic acid. The full name is rarely needed, but its effects can be seen across a strawberry as it enlarges. Auxin changes which genes are active inside nearby cells. Some of those cells divide, increasing their number. Others expand as water enters, and their walls loosen enough to stretch. The hormone also supports the development of vascular tissue, the narrow transport pathways carrying water, minerals, sugars, and chemical messages through the plant. Together, these changes keep the receptacle in an active period of growth. The signal is spread across the fruit because hundreds of developing achenes sit at different points on its surface. Each one contributes to the growth of the tissue close beneath it. When fertilization is even, auxin-related signals are distributed across most of the receptacle. The berry can then widen near its shoulders and fill out toward its tip with a fairly regular shape. A mist group of pistils creates a different result. Several ovaries may remain small because they receive too little viable pollen or because fertilization failed. Their achenes stay pale, hard, and poorly developed. The patch beneath them receives less of the normal growth signal. Tissue nearby continues expanding, while the affected area remains tighter and lower. A shallow groove, flattened side, or uneven shoulder may gradually appear. Small undeveloped achenes can often be seen clustered within the sunken area. A misshapen strawberry, therefore, carries a visible record of how development varied across one flower. Poor insect activity can contribute when pollen is spread unevenly, although several other causes can produce the same pattern. A late spring frost may damage stigmas or ovaries while the flower is open. Thrips and other small insects can feed on delicate flower tissue. Heavy rain may wash pollen away, while unusually cold weather can slow the growth of pollen tubes before they reach the ovules. Physical injury to a cluster of pistils can also interrupt normal development. For this reason, an irregular berry cannot provide a simple measure of how many bees visited the plant. The connection between achenes and receptacle growth became especially clear in classic plant development experiments. researchers removed developing achenes from part or all of a young strawberry. Growth slowed strongly in the tissue beneath the cleared area. When an auxin solution was placed onto that tissue, some growth could be restored. The receptacle responded to the chemical after the natural source had been taken away. Areas that retained their achenes continued developing more normally, creating a clear boundary between strongly growing tissue and the sections where the small fruits had been removed. These experiments help show that the achenes act as small control centers distributed over the fruit surface. Their developing seeds and surrounding tissues release signals that influence the cells below. Auxin works within a larger network and plays a major role while the young strawberry is still green and expanding. Later, changing hormone levels will help shift the fruit toward red pigment. Its walls will soften, and its recognizable aroma will begin to form. For now, the receptacle is being built region by region under the guidance of hundreds of small fruits. Chemical instructions alone cannot produce new tissue. Every dividing cell needs water and dissolved minerals. Expanding walls require sugars and other carbon compounds made through photosynthesis. Those supplies must arrive from a compact plant body resting close to the soil beneath the leaves. The supplies entering a young strawberry travel through a plant that may stand only 20 or 30 centimeters above the ground. At the center of that plant is a thick structure called the crown. The crown sits at the soil line, where the roots disappear underground and the leaves rise into the light. Botanically, it is a short, compressed stem. Its sections are packed so closely together that it never develops the long spaces seen between the leaves of many upright plants. A healthy crown can survive for several growing seasons. Stored carbohydrates inside its tissues help the plant endure winter dormancy and begin growing again when spring temperatures rise. Carbohydrates are energy-rich compounds made from carbon, hydrogen, and oxygen. Sugars are one familiar form, while starch provides a more compact way for the plant to store part of that energy. Several kinds of growth emerge from tightly grouped points around the crown. Leaf stalks extend upward and outward. Flower stems lift clusters of buds above the foliage. Roots descend from the crown's lower surface, while horizontal runners may later stretch across the ground. Older plants can also develop side crowns, sometimes called branch crowns. Each side crown creates another growing center capable of producing its own leaves and flower cluster. A plant with several healthy crowns may therefore carry more flower stems during a later season. The number cannot increase without limit. Crowded crowns compete for light and water, and dense foliage can trap moisture around flowers and fruit. Above the crown, a typical strawberry leaf is trifoliate. This means that one leaf is divided into 3 separate leaflets. Each leaflet has a toothed edge and a network of veins that carries water into the tissue while transporting manufactured sugars away. Inside the leaf cells are chloroplasts, tiny structures containing the green pigment chlorophyll. Chlorophyll absorbs energy from sunlight. During photosynthesis, that energy helps combine carbon dioxide from the air with water drawn from the soil. The process produces sugars that can be used immediately or transported to growing parts of the plant. Young green strawberries perform a limited amount of photosynthesis of their own. Their surface contains chlorophyll before ripening removes much of the green color. Mature leaves still provide most of the sugar imported into an enlarging fruit. Those sugars move through phloem, the part of the vascular system that carries dissolved food from producing tissues toward places where it is needed. Water and minerals arrive mainly through xylem, a second transport tissue extending upward from the roots. Below the crown lies a dense network of fibrous roots. The finest branches are often called feeder roots because their large combined surface area absorbs much of the plant's water and dissolved nutrients. Many active feeder roots remain within the upper 15 centimeters of soil, or roughly 6 inches. Some larger roots can reach farther downward when the soil is deep, loose, and well aerated. This shallow pattern gives the plant quick access to nutrients near the surface, where decaying organic matter and fertilizer are often concentrated. It also leaves strawberries sensitive to drying. During hot weather, the upper soil layer may lose moisture rapidly through evaporation. If water becomes scarce while the receptacle is enlarging, its cells cannot maintain normal expansion. Fruit may remain smaller, and prolonged stress can reduce flower production or damage shallow roots. Nearby weeds increase the pressure by drawing from the same upper layer of soil. Too much water creates a different problem. Roots need oxygen for cellular respiration, the process that releases usable energy from stored sugars. When standing water fills the air spaces between soil particles, oxygen reaches the roots more slowly. Waterlogged conditions can weaken root function and favor diseases caused by organisms such as Phytophthora, whose swimming spores move especially well through wet soil. The crown must continually divide its available resources. Some carbohydrates support new leaves and repair roots. A large share may flow into swelling fruit during harvest. Other reserves remain stored for dormancy, while hidden buds begin preparing future flowers. The same compact stem can also direct energy into a long runner, carrying part of the plant's resources away from the crown toward a new patch of soil. From the crown, a slender green stem begins extending sideways across the soil. It may pass beneath a leaf, cross an open patch of mulch, and continue beyond the parent plant's outer edge. This horizontal stem is called a stolon. Gardeners usually call it a runner. Both names describe the same structure. Unlike an upright flower stalk, the runner remains close to the ground and carries the plant's growth into nearby space. Small joints called nodes appear along its length. A node is a point where a stem can produce new organs. When one of these nodes rests against moist soil, a cluster of leaves begins rising from its upper side. Fine white roots emerge beneath it and press downward between particles of sand, clay, and decaying plant material. These are adventitious roots. The word adventitious means that they develop from stem tissue, rather than branching from an older root. Within a short time, the node begins forming a new crown. This young crown is called a daughter plant. Its first roots may be only a few millimeters long, leaving it dependent on the parent strawberry. Water can pass through the runner from the established crown. Dissolved minerals absorbed by the mother plant's roots may travel through the same connection. Sugars made in mature leaves can also move toward the developing daughter, while its own leaf area remains small. The connection gives the new plant time to anchor itself and build a working root system. As the daughter grows, its leaves expand and begin producing more of their own sugar. New roots explore the upper soil layer. The runner between parent and daughter may eventually dry, weaken, or break. By then, the daughter can continue as an independent strawberry plant. A strong mother plant can produce several runners during a favorable growing season. Some runners form more than one daughter plant because a second node continues growing beyond the first. Over several months, one crown can become the center of a spreading patch. This method of reproduction is known as vegetative reproduction. A new plant is produced from the parent stem tissue without requiring a flower to form seeds. The daughter receives nearly the same genetic information as the mother plant. This makes it a clone. Seed-grown strawberries develop differently. Pollen and ovules combine genetic material from two reproductive cells. Their offspring receive a newly mixed set of gene versions, so seedlings can vary in fruit size, flowering time, disease resistance, and many other traits. Clonal propagation preserves the useful combination already present in a named cultivar. A plant sold as Albion, Chandler, or Honioye can be multiplied through runners while keeping the traits that led growers to select it. Small differences can still appear among clones. A mutation may occur in one growing point as cells divide. These changes are called somatic mutations because they arise in ordinary plant tissue. Soil, light, temperature, and water supply also change how genetically similar plants grow. Nurseries use clonal propagation to produce large numbers of plants with predictable traits. Healthy mother plants may be tested for viruses and maintained under protected conditions. Their runners are rooted, separated, and grown until they are large enough for sale. Clean starting material matters because clonal propagation can preserve pathogens along with useful genes. Viruses may remain inside plant tissue and move into each new daughter. Fungal organisms can persist around crowns, roots, or nursery soil. A hidden infection may then travel from a propagation field into many farms. Certified nursery programs reduce this risk through plant inspection, testing, sanitation, and carefully managed stock. Fruit growers treat runners differently according to the production system. In a June-bearing matted row, daughter plants are encouraged to root within a strip of soil. Their crowns fill the row and create many flowering points for the following year. In annual plasticulture, plants grow through holes in sheets of plastic mulch. Runners spreading across the plastic are often removed because they cannot root easily and continue consuming sugar. Day-neutral growers may also cut runners throughout the season. This keeps foliage more open, improves air movement, and directs a larger share of the plant's resources toward repeated flowers and fruit. The same crown can therefore invest in spreading across the ground or building a harvest above it. Its choice is strongly influenced by the changing duration of daylight and the temperature surrounding its leaves. Inside the strawberry's leaves, light is being measured. Plant cells contain light-sensitive proteins that respond to changes in brightness and to the colors within sunlight. One important group is called phytochrome. These proteins help the plant compare the length of daylight with the darkness that follows. The full duration of daylight within a 24-hour cycle is called the photoperiod. As summer passes and nights become longer, the changing photoperiod alters chemical signals moving from the leaves toward the crown. Genes inside the crown respond by changing which structures its growing points begin to form. A bud that might have produced another runner can begin developing into a flower bud instead. This process remains hidden among tightly packed layers of young tissue. A berry harvested in June may therefore begin its development during the previous August or September. Many traditional June-bearing strawberries are known as short-day plants. Their flower initiation is encouraged when days shorten. although suitable temperature is also required. The plant does not count calendar dates. It responds to the environmental pattern surrounding it. A stretch of cool autumn weather combined with longer nights can favor flower bud formation. Unusually warm conditions may delay or weaken that response, even when daylight has become shorter. Within each hidden bud, the first parts of a future flower cluster begin forming. The plant may establish the number and position of blossoms months before any white petals become visible. Some of those buds will later produce primary flowers, which commonly form the largest berries. Others will become the smaller secondary and tertiary blossoms arranged around them. As temperatures continue falling, visible growth slows. Leaves may remain low against the soil, while the crown enters dormancy. Dormancy is a controlled period of greatly reduced growth that helps a perennial plant survive an unfavorable season. Strawberry crowns store carbohydrates in their cells before winter. These reserves support basic metabolism during the cold months and provide energy when growth resumes. Cold exposure also helps many cultivars complete dormancy. This seasonal requirement is often described as chilling. The needed amount varies among cultivars and may be counted as hours spent below a chosen temperature. A strawberry bred for a cold northern climate may respond differently from one developed for a mild coastal winter. Cold can provide a useful signal, while still becoming damaging at greater extremes. An unprotected crown may be injured when its tissues freeze deeply or experience repeated freezing and thawing. Flower buds hidden inside the crown can also be damaged before spring begins. A layer of straw mulch traps pockets of insulating air around the plants. Snow can provide additional protection by keeping the crown's temperature more stable than the exposed air above it. When spring warmth returns, stored reserves help the crown produce new roots and leaves. The flower clusters prepared during autumn begin stretching upward. A June-bearing bed may then produce most of its crop during a concentrated period lasting several weeks. Day-neutral cultivars follow a different schedule. Their flower initiation is less tightly controlled by day length, allowing them to form blossoms through a much larger part of the growing season. Cultivars such as Albion and Seascape can produce repeated waves of flowers and fruit when temperatures remain suitable. Many day-neutral strawberries perform well when average conditions stay between about 7 and 29 degrees Celsius, or 45 and 85 degrees Fahrenheit. Day neutral does not mean unaffected by the environment. High temperatures can reduce flower formation and pollen quality. Heat also speeds fruit development, which may shorten the time available for sugars and aroma compounds to accumulate. Very cool weather slows flowering and lengthens the period between bloom and harvest. The older horticultural term, everbearing, is often used for plants that produce one crop in spring and another later in summer or early autumn. Modern-day neutral cultivars may flower more continuously, although their production still rises and falls with temperature, plant age, and resource supply. Every flowering pattern grows from a particular way of reading the seasons. Those different responses were shaped long before named cultivars appeared, as wild strawberries adapted to woodland shade, exposed coastlines, mountain slopes, and short northern summers. Along a cool woodland edge, a wild strawberry plant may be almost hidden beneath taller grasses and fallen leaves. Its fruit is often smaller than a garden strawberry, sometimes no wider than a fingertip. The scent can still be strong enough to notice before the berry is seen. Wild members of the strawberry genus, called Fragaria, grow across a wide range of habitats. Some occupy open forests where broken sunlight reaches the ground. Others spread along meadow borders, mountain slopes, sandy dunes, or coastal vegetation exposed to wind and salt spray. Each population has been shaped by the conditions surrounding it. Cold winters favor plants whose crowns and flower buds survive freezing. Dry ground rewards root systems that use water carefully. Coastal environments place pressure on leaves and roots that must tolerate salt carried through soil or sea air. One of the most widespread species is Fragaria vesca, commonly called the woodland strawberry. It occurs across broad parts of Europe, Asia, and North America. Its bright red fruit is usually small and narrow, with a fragrance that can seem unusually concentrated for its size. Woodland strawberries often grow in clearings, along paths, and near forest margins, where they receive more light than plants beneath a closed canopy. Fragaria vesca is generally diploid. Diploid means that its cells carry 2 complete sets of chromosomes. Each set contains 7 chromosomes, giving the plant 14 in total. Several alpine forms have been selected from this species. Some produce few runners, while others form none at all. Instead of quickly spreading sideways, they keep their growth concentrated around one crown and may flower over a long season. Another European and Asian species is Fragaria viridis, often called the green strawberry. Its ripe fruit may remain pale red, greenish, or partly white. The flesh can be firm, and the berry sometimes separates from its calyx with a small snapping motion. Farther west in Europe grows Fragaria muscata, the musk strawberry. This species is generally hexaploid, meaning its cells hold 6 chromosome sets. Its fruit is known for a deep aroma that may seem musky, spicy, or wine-like. Musk strawberries were once grown more widely in European gardens, especially before the modern garden strawberry became dominant. Their flowers also reveal how varied strawberry reproduction can be. Some wild species carry flowers with working male and female parts. Other populations include separate female plants and pollen-producing male plants. A flower's appearance alone may therefore give only part of the story. Fruit production depends on whether its reproductive structures function and whether compatible pollen reaches the stigmas. Across North America, Fragaria virginiana grows from parts of Canada through much of the United States. It tolerates cold winters and produces small, flavorful berries. This species is octoploid, with 8 chromosome sets and 56 chromosomes. Its genetic traits later became important in the ancestry of cultivated strawberries. Along Pacific coastlines grows another octoploid species, Fragaria chiloensis. It is often called the beach strawberry or Chilean strawberry. Populations occur along western North America and continue far south into Chile. Thick leaves, low growth, and sturdy runners help some coastal forms withstand wind, shifting sand, and salt exposure. Certain Chilean plants also produce fruit much larger than that of many other wild strawberries. Wild fruit size varies greatly, yet size alone says little about how advanced a plant is. A small berry may contain a powerful aroma profile, strong disease resistance, or an ability to survive conditions that would damage a larger garden cultivar. Commercial breeding programs still return to wild populations for useful traits, One species may carry resistance to a root pathogen. Another may flower under warmer conditions or survive colder winters. A population from dry ground may hold gene variants that improve heat or drought tolerance. These qualities developed through local selection over many generations. Plants leaving more offspring under a particular climate gradually made useful traits more common in that place. The wild strawberry lineage, therefore, contains a collection of different solutions to different environments. Some spread rapidly through runners. Others remain compact. Their fruits vary in firmness, color, and fragrance, while their flowers use several reproductive arrangements. Beneath those visible differences lies an even larger divide. Certain species carry 2 chromosome sets. Others hold 6 or 8, creating genomes with repeated copies of thousands of genes. The cultivated strawberry would eventually emerge from that deeper genetic complexity, carrying 56 chromosomes inside nearly every cell. The woodland strawberry organizes its 14 chromosomes into 2 complete sets of 7. One set comes from the pollen parent, while the other comes from the plant that produced the ovule. Chromosomes are long packages of DNA carrying thousands of genes. 2 matching sets allow related chromosomes to pair when reproductive cells are made. This condition is called diploidy. Humans are diploid as well, although our cells contain 46 chromosomes arranged in 23 pairs. Many plants follow a more complicated pattern. Sometimes an entire chromosome set is copied. In other cases, chromosome sets from related species become combined through hybridization. A plant with 4 sets is tetraploid. 6 sets make it hexaploid, and 8 sets make it octaploid. The broad name for these conditions is polyploidy, meaning that a cell carries more than 2 complete chromosome sets. Polyploidy is common among flowering plants. Wheat, potatoes, cotton, and several familiar fruits carry duplicated or combined genomes. These extra sets can appear when reproductive cells are formed with more chromosomes than usual, or when chromosomes double inside a young hybrid. The cultivated garden strawberry, Fragaria cross and anassa, is octaploid. Nearly every ordinary body cell contains 56 chromosomes, arranged as 8 sets of 7. That total is 4 times the chromosome count of the diploid woodland strawberry. The 8 sets carry related but distinct ancestral histories. The cultivated genome contains several subgenomes. A subgenome is one ancestral chromosome set, or group of related sets, preserved inside a larger hybrid genome. This arrangement is called allopolyploidy. The prefix allo means different, because the chromosome sets came from different ancestral lineages. Long before strawberries entered European gardens, older wild species crossed and their genomes combined. Further chromosome duplication allowed those hybrid descendants to reproduce successfully. This deep assembly took place over evolutionary time, probably beginning more than 1 million years before modern cultivation. By the time the immediate wild parents of the garden strawberry appeared in the Americas, both already carried the octaploid condition. The modern cross therefore joined 2 plants that each possessed 56 chromosomes. Their offspring inherited a layered genetic system, assembled through much older events. Researchers can compare DNA sequences across modern species to trace those ancestral contributions. Some parts of the history remain uncertain. Several proposed diploid ancestors are closely related, and later gene exchange blurred the boundaries among their descendants. New genome maps continue to refine which ancient lineage supplied each chromosome region. One subgenome, related to Fragaria vesca, the woodland strawberry, appears especially influential. Across many tissues, genes from this subgenome are expressed more strongly or more often than related copies from some of the other ancestral sets. Gene expression means that a gene is being used to make RNA and often a protein. A dominant subgenome can therefore contribute a larger share of the plant's active instructions, while the other copies remain present. Duplicated genes create several possible outcomes. One copy may preserve the original function, while another gradually takes on a more specialized task. A damaged version can sometimes be masked because another related copy still works. Some copies become less active over generations, while others respond differently in roots, flowers, or ripening fruit. This redundancy gives evolution more material to modify. It also makes strawberry breeding difficult. A trait, such as firmness, may be influenced by several related gene copies spread across different chromosome sets. Changing one region may produce only a small effect because other copies continue performing similar work. Breeders must often track patterns across the full genome instead of searching for one simple switch. Inside a single strawberry cell, ancient chromosome mergers remain active in growth, disease resistance, flowering, color, and aroma. That genetic structure was already in place when human travel carried two distant American species into the same European gardens during the early 18th century. One octoploid strawberry lineage spread across much of North America. This species, Fragaria virginiana, grew from parts of Canada through broad regions of the United States. Its berries were usually smaller than modern cultivated strawberries, yet many carried a rich aroma and strong flavor. The plants also survived cold continental winters, where frozen ground and short growing seasons favored crowns able to protect their hidden buds. By the early 17th century, living plants had crossed the Atlantic and entered European gardens. Gardeners valued their fruit, though the species alone did not yet produce the large strawberries familiar today. A second octoploid strawberry grew thousands of kilometers away along the Pacific edge of the Americas. Fragaria chiloensis occupied coastal habitats from western North America into South America. Some populations grew close to beaches where low leaves faced wind, shifting sand, and salt carried inland from the ocean. In Chile, indigenous communities had already selected and cultivated forms with unusually large fruit before Europeans collected them. Several Chilean strawberries were pale red or nearly white when ripe. Their receptacles could grow much larger than those of many European and North American wild species. In 1712, a French military engineer named Amédée François Fraisier reached the Chilean coast during a voyage connected with mapping and military observation. He examined local crops and collected several strawberry plants. Keeping them alive during an ocean crossing required constant care. Fresh water was limited aboard the ship, and the voyage back to Europe lasted for months. Fraisier returned to France in 1714 with a small number of surviving plants. His surname sounds remarkably close to the French word for strawberry, fraise. The resemblance is a historical coincidence. The French word existed long before his voyage and did not come from his family name. The transported Chilean plants created a practical problem once they began growing in France. Most were female plants. Their flowers could receive pollen and produce seeds, but they supplied little or no useful pollen of their own. Planted together without a compatible pollen source, they flowered while producing few normal berries. Gardeners eventually placed them near other strawberry species. Pollen carried by insects reached the Chilean flowers from nearby plants, allowing some fruit to form. Among the possible partners was the North American Fragaria virginiana, which had already been growing in European collections for decades. Both species carried 8 chromosome sets. That shared octoploid structure helped their chromosomes function together in hybrid offspring. The plants had remained separated by enormous geographic distance in the Americas. One lineage occupied much of a northern continent. The other included coastal populations extending into Chile. European transport placed them within the flight range of the same bees. Cross-pollination combined the larger fruit associated with Chilean plants with useful traits from the North American species, including fertility, flavor, and adaptation to colder conditions. The first hybrids were variable. Some produced disappointing fruit or weak plants. Others grew vigorously and formed berries larger than those of many strawberries already present in European gardens. Growers saved useful plants and multiplied them through runners. Further crosses gradually produced more dependable combinations. This new cultivated strawberry became known as Fragaria, cross ananassa. The cross in its scientific name marks the species as a hybrid. The word ananassa referred to an aroma that some early observers compared with pineapple. Hybrid development became especially important in French gardens and spread through nearby parts of Western Europe during the 18th century. The exact modern form did not appear through one carefully planned crossing on a recorded day. It emerged through repeated pollination, selection, and clonal propagation across gardens containing several strawberry species. The cultivated garden strawberry is therefore only about 3 centuries old. The 8 chromosome sets inside it preserve a history reaching back more than 1 million years, while the crop itself arose after ships moved 2 compatible American plants beyond the ranges where they had evolved. Their meeting produced the biological foundation of nearly every large strawberry now grown around the world. Understanding the parentage required botanists to watch which flowers produce pollen, which plants form fruit, and which traits appeared again in their descendants. Across 18th century European gardens, several kinds of strawberry grew close together. Some carried tiny dark red berries. Others produced pale fruit, broad leaves, unusual runners, or flowers that opened without forming a normal harvest. Hybrids appeared among them, although their parentage was difficult to trace. The plants had arrived from different continents, and the science of inheritance had no modern language yet. Antoine-Nicolas Duchesne entered this confusion with careful eyes and years of patient observation. He was born in France in 1747 and lived until 1827. Duchesne began studying strawberries while still young, working with plants connected to the royal gardens at Versailles and other French collections. Versailles offered more than formal paths and carefully shaped trees. Its cultivated grounds held living specimens gathered from Europe and distant territories, allowing related plants to grow within the same garden. A botanist could compare their leaves, flower structures, runner habits, and fruit over several seasons. Duchenne paid close attention to reproduction. He recorded which plants released useful pollen and which flowers developed receptacles after nearby varieties began blooming. He watched how runners preserved the features of one parent plant. Seeds gave him a different kind of evidence because each seedling could display a fresh mixture of inherited traits. One plant might carry the leaf shape of one parent and the fruit color of another. A later generation could reveal features that had been hidden in the first cross. Repeated comparisons allowed patterns of inheritance to appear gradually. Strawberry flowers made this work especially complicated because the genus contains several reproductive systems. Many cultivated strawberries have hermaphroditic flowers, meaning that one flower carries working male and female structures. Certain wild populations include female plants whose flowers can receive pollen, while producing little useful pollen of their own. Other plants function mainly as males. Some species show additional variation in how strongly each structure works. These differences helped explain the poor fruiting of the Chilean strawberries carried to France. Their flowers open normally, yet many of the imported plants were female. Fruit production improved when compatible pollen arrived from another strawberry growing nearby. Duchenne could infer relationships by controlling or observing those pollen sources. When a plant produced offspring with a combination of traits associated with two neighboring forms, hybrid ancestry became a strong explanation. The same conclusion gained support when similar crosses repeatedly gave similar results. His work depended on visible evidence. Flowers could be covered or isolated to limit unwanted pollen. Selected plants could be placed together. Offspring could be raised, labeled, and compared with their parents. Every season added another layer of information. Duchenne had no microscope capable of showing chromosomes. DNA would not be identified as the material of inheritance until much later. His tools were living plants, detailed notes, and comparisons repeated across generations. In 1766, when he was about 19 years old, Duchenne published Histoire naturelle des fraisiers, translated as Natural History of Strawberries. The book organized the strawberry forms known to him and described their growth, reproduction, and relationships. It became an important early study of plant variation and hybridization. Duchenne recognized that the emerging garden strawberry had a hybrid origin involving the large Chilean strawberry and a compatible North American form. That conclusion helped bring order to plants that gardeners had already been crossing, selecting, and passing between estates. A useful hybrid could now be understood as a combination of inherited traits. Growers could save seedlings with larger fruit, stronger flowering, or improved fertility. Runners then allowed a successful plant to be copied without mixing its genes again. The garden accident had become material for directed breeding. Over the following centuries, thousands of crosses would reshape berry size, firmness, harvest timing, disease resistance, and flavor, gradually producing the strawberries found in fields and markets today. A strawberry breeder begins with two parent plants chosen for useful qualities. One may produce large, firm fruit, while the other carries strong aroma or resistance to a common disease. Pollen is collected from one flower and placed onto the stigmas of the other. The resulting achenes contain seeds with newly mixed genetic combinations. Each seedling is different because it receives its own selection of gene versions from the parents. These gene versions are called alleles. A single cross may produce hundreds or thousands of seedlings. Breeders plant them in fields or greenhouses and wait for the first flowers and fruit. Most seedlings are removed after evaluation. A small number may continue into later trials, where their performance is measured across several seasons and locations. Fruit size is only one part of that evaluation. Researchers record total yield, berry shape, external color, and the color visible after the fruit is cut. Firmness may be measured by pressing a probe into the receptacle and recording the force required. Sweetness is often estimated through soluble solids, while acidity is measured separately. Aroma, flowering pattern, resistance to pathogens, and useful shelf life also influence whether a plant survives the selection process. The qualities growers valued have changed with the way strawberries are produced and sold. Early European gardeners often focused on berry size, unusual color, and visible appearance. As commercial farms expanded, a successful cultivar also needed to produce a dependable crop and ripen at a useful time. Fruit shipped over long distances required enough firmness to survive picking, packing, cooling, and transport. Modern breeding programs may also select for day-neutral flowering, which allows plants to produce fruit across a longer season. Large strawberries can develop through more than one growth pattern. Some genotypes produce a greater number of cells within the receptacle. Others enlarge each cell more strongly as water and sugars enter. Many large cultivars combine both processes. The final size still depends on pollination, crop load, temperature, and water supply. So genetics establishes a range rather than one guaranteed result. Wild strawberries remain valuable sources of traits that may be rare in cultivated plants. A wild population exposed to a local fungus for many generations may carry alleles that slow infection. Plants growing near a hot coastline may hold useful forms of genes involved in heat tolerance or water use. Breeders can cross that wild material with cultivated strawberries and then repeatedly cross the descendants back to commercial parents. This process is called introgression. Its purpose is to move a useful region of DNA into a cultivated background, while keeping as many desirable crop traits as possible. The process can take several generations, because wild plants may also contribute small fruit, low yield, or flowering habits poorly suited to farms. Modern genetic markers help breeders track the desired chromosome region, while unwanted regions are gradually reduced. Repeated selection also leaves recognizable patterns in the genome. When a valuable allele becomes common because breeders continually choose plants carrying it, nearby DNA may rise in frequency along with it. This pattern is called a selective sweep. Genetic diversity around the favored region becomes lower because many successful plants inherited the same stretch of chromosome from a shared ancestor. Strong selection creates useful uniformity, while narrowing the genetic options available in that region. Breeding goals can also pull the fruit in several directions at once. Very firm tissue may travel well, yet excessive firmness can produce an unpleasant texture. A plant carrying a heavy crop must divide its sugars among many berries, which can lower sweetness when leaf production and sunlight do not supply enough carbon. Selection for rapid ripening may shorten the period during which aroma compounds accumulate. Commercial strawberries still cover a wide range of flavor. Cultivar, harvest maturity, climate, water supply, storage temperature, and time after picking all influence sweetness and fragrance. A berry harvested fully red near the place where it was grown may taste different from the same cultivar picked earlier for a long journey. Once breeders identify a promising seedling, they preserve it through clonal propagation. Runners produce daughter plants carrying nearly the same genome. Tissue culture can also multiply plants from small pieces of clean-growing tissue under sterile conditions. These methods allow one selected individual to become thousands of plants with predictable traits. Every cultivar remains dependent on the same brief transformation. During a few weeks on the plant, its hard green receptacle must activate the internal program that creates red pigment, sweetness, aroma, and softness. Near the end of its month-long development, a strawberry remains firm, green, and closely connected to the plant. Its achenes have already helped guide the receptacle through its main period of enlargement. During that early stage, developing seeds and achene tissues produced auxin, a group of chemical signals that encourage nearby cells to divide and expand. As the seeds approach maturity, auxin's influence begins to decline. Growth slows, and the receptacle enters a different kind of activity. Many of the cells that now form the fruit are already in place. Their walls, pigments, stored sugars, and aroma compounds will be altered during the remaining days before harvest. The change first passes through a pale stage. Chlorophyll begins disappearing from the receptacle, causing the green surface to fade toward white. At this point, another plant hormone becomes especially important. It is called abscisic acid, usually shortened to ABA. The name can sound misleading because abscisic acid was once linked with leaves and fruit dropping from plants. Researchers now know that it has much wider roles. It helps plants respond to drought, influences seed dormancy, and coordinates important parts of strawberry ripening. ABA begins accumulating strongly in the receptacle as the fruit changes from white to pink and then red. Cells detect the hormone through special receptor proteins. This starts a chain of chemical activity inside the cell, changing which genes are switched on and how strongly they operate. Some of those genes help manufacture anthocyanins, the pigments that color the ripening strawberry. Others affect the movement and use of sugars. Additional pathways loosen the fruit's cell walls and increase production of molecules that later contribute to its scent. A BA acts within a network rather than working alone. Jasminates, a group of hormones often involved in plant defense and development, can influence parts of ripening. Gibberellins are more active during earlier growth and interact with pathways controlling cell expansion. Brasinosteroids, hormones that help regulate growth and stress responses, may also affect the timing and strength of ripening changes. Ethylene remains present as well. This gas-like plant hormone has a central role in fruits such as bananas and tomatoes. Those fruits are described as climacteric. As ripening begins, their respiration rises sharply and ethylene production increases. Respiration is the process through which cells break down sugars to release usable energy. A mature green banana can therefore continue through a major ripening program after it has been removed from the plant. Its peel becomes yellow, starch is converted into sugar, and the flesh softens over several days. Strawberries are commonly classified as non-climacteric fruit. They do not produce the same large respiratory surge after harvest. Ethylene may still influence certain tissues and stages, though it does not take control of the full process in the way seen in a banana or tomato. A strawberry can deepen slightly in color after picking. Some remaining pigment reactions continue, especially when the fruit was already close to red. Its cells also continue respiring, softening, and losing water. The most important gains in sweetness and aroma depend heavily on remaining attached to the plant. Leaves continue sending sugars into the receptacle through the phloem until the fruit is picked. Aroma pathways also remain active during the final stages of ripening. A pale strawberry removed early cannot rebuild that missing period simply by resting on a kitchen counter. It may become softer and appear somewhat redder, while its flavor stays weak. Commercial growers must therefore choose harvest timing carefully. Fruit intended for nearby sale can remain attached until it reaches a deeper stage of ripeness. Berries facing a longer journey may be picked while still firm enough to survive handling, though removing them too early lowers their final eating quality. Within each fruit, the timing reflects a gradual chemical handoff. Auxin's early support of enlargement gives way to rising ABA activity and a wider network of ripening signals. The first clear record of that shift appears across the receptacle as green and white tissue begins filling with red pigment. At the pale stage, the strawberry has already reached much of its final size. Its receptacle feels firm, and the surface may appear cream-colored beneath a faint green cast. Over the following days, patches of pink begin forming around the achenes and across areas exposed to light. The color gradually spreads until the outer tissue becomes fully red. Growers often describe these stages as green, white, turning, and ripe. Each visible stage reflects changes taking place inside millions of receptacle cells. While the young fruit is green, many of those cells contain chloroplasts. A chloroplast is a small structure that holds chlorophyll, the pigment used to absorb light during photosynthesis. The green receptacle can manufacture a modest amount of sugar for itself, although mature leaves still provide most of the carbon entering the fruit. As ripening advances, chlorophyll is broken apart. The green color fades, revealing pale tissue before enough red pigment has accumulated to cover it. Chloroplasts belong to a larger family of cell structures called plastids. During ripening, their internal membranes and chemical activity change. They lose much of the machinery associated with photosynthesis and become increasingly involved in storing pigments and other compounds. At the same time, rising ABA signals activate genes that control the production of anthocyanins. Anthocyanins are water-soluble pigments belonging to a larger chemical group called flavonoids. Plants use flavonoids in coloration, protection from ultraviolet light, and responses to environmental stress. Their construction begins with carbon compounds made through photosynthesis. A sequence of enzymes then modifies those starting materials, one reaction at a time. This connected series of reactions is known as the flavonoid pathway. Several enzymes build the basic pigment structure. Later enzymes attach sugar molecules or make other small changes that improve stability and affect color. The most abundant anthocyanin in many cultivated red strawberries is pelargonidin 3-glucoside. Its chemical structure produces much of the fruit's bright scarlet appearance. A related pigment called cyanidin 3-glucoside is usually present in smaller amounts and can add deeper red tones. The balance between these compounds differs among cultivars. Pigments formed in the cell fluid are moved into vacuoles. A vacuole is a large, liquid-filled compartment that occupies much of the inside of a mature plant cell. It stores water and dissolved chemicals, while helping the cell maintain internal pressure. As anthocyanins collect inside these compartments, the tissue becomes visibly red. The acidity inside a vacuole influences the exact shade that appears. Pigment structure also matters, along with interactions involving other dissolved compounds. Gene activity controls how quickly this color system develops. Proteins called transcription factors act as regulators. They attach to selected regions of DNA and influence whether nearby genes are used strongly, weakly, or hardly at all. One important strawberry regulator is called FAMYB10. It helps activate several genes needed for anthocyanin production. When this regulator is highly active during ripening, pigment-making enzymes increase and red color spreads through the receptacle. Natural mutations can weaken this process. Some white-fruited strawberries carry changes affecting pigment regulation, including versions of the FAMYB10 system. Their receptacles can become sweet and soft while producing little red anthocyanin. Pale cultivars show that ripening contains several connected processes whose timing can vary. Conditions around the plant also shape the final color. Light often encourages anthocyanin accumulation, so shaded areas may develop more slowly. Cool nights can support strong pigmentation in some cultivars, while extreme heat may reduce pigment stability or alter gene activity. Water stress sometimes increases certain protective compounds, although severe stress restricts growth and damages fruit quality. Cultivar genetics remains a major influence, determining the range within which those environmental effects operate. A fully red surface is therefore a useful sign that ripening has advanced. It does not guarantee that every berry contains the same sugar concentration or aroma mixture. Those parts of the fruit's transformation depend on dissolved sugars, organic acids, and compounds that begin escaping into the air as the receptacle approaches full ripeness. Beneath the red surface, dissolved sugars accumulate in the water held inside the receptacle's cells. 3 sugars provide most of a ripe strawberry sweetness: glucose, fructose, and sucrose. Glucose and fructose are simple sugars that can be used directly during cellular respiration. Sucrose is built from one glucose unit joined to one fructose unit. The leaves manufacture sucrose during photosynthesis and load it into the phloem, the plant tissue that carries dissolved food toward growing fruit. Once sucrose enters the receptacle, enzymes may split it into glucose and fructose. Other enzymes can rebuild sucrose or direct part of the sugar into different chemical pathways. Fruit cells also consume some of this supply to release energy for pigment production, aroma formation, and the many reactions involved in ripening. The final sugar mixture varies among cultivars. Some strawberries contain relatively more fructose, which tastes sweeter to people than the same amount of glucose. Others carry a larger share of sucrose. Temperature, leaf health, sunlight, crop load, and harvest timing all influence how much sugar reaches each berry. Water supply changes the concentration as well. A strawberry receiving abundant water near harvest may expand quickly as its cells fill. The total amount of sugar can still increase, while the larger volume of water makes that sugar more diluted. Dry conditions may concentrate dissolved solids for a time, although serious water stress reduces photosynthesis and limits fruit growth. sugar forms only one side of strawberry flavor. The receptacle also contains organic acids, with citric acid usually making the largest contribution. Malic acid adds part of the tartness, while smaller quantities of other acids help shape the overall taste. These compounds stimulate sour-sensitive receptors on the tongue. Their presence can make sweetness seem brighter and more defined. A fruit with moderate sugar and low acidity may taste mild. Another berry with the same sugar concentration can seem sharper because it contains more acid. Growers and researchers often estimate dissolved solids with a refractometer. This small instrument measures how strongly a drop of juice bends light. The result is expressed in degrees Brix. In a simple sugar solution, 8 degrees Brix would represent roughly 8 grams of sucrose in 100 grams of liquid. Strawberry juice contains acids, minerals, and other dissolved substances along with sugars. A reading of 8 degrees Brix therefore does not mean that exactly 8% of the fruit is sugar. It remains a useful estimate for comparing berries harvested from the same crop or tested under similar conditions. Commercial quality guidance often treats about 7% soluble solids as a practical minimum. A strawberry reaching that level can still taste disappointing when its acidity is poorly balanced or its aroma is weak. Breeders and growers may compare soluble solids with titratable acidity. This measurement estimates how much base must be added to neutralize the acids in a known amount of juice. It reflects the fruit's total neutralizable acid more completely than a simple acidity reading based only on hydrogen ions. The ratio between soluble solids and titratable acidity often gives a better clue to flavor than either number alone. Taste can even change across one strawberry. The pointed tip often contains a higher concentration of soluble sugars than the broad shoulder beneath the calyx. Acid levels may be distributed more evenly. A bite from the tip can therefore seem sweeter than a bite taken close to the green cap. Ripening pattern, vascular supply, and differences in tissue development may all contribute to this gradient. Serving temperature changes the experience again. A refrigerator keeps strawberries near 4 degrees Celsius in many homes. At that temperature, fewer volatile aroma molecules escape from the fruit into the air. Taste receptors still detect sugars and acids, yet the reduced scent can make the berry seem less sweet and less intense. Allowing the fruit to warm slightly increases evaporation from its surface and from the juice released during chewing. 2 equally red strawberries can therefore produce very different flavors because sweetness is assembled from sugar concentration, acidity, aroma, texture, and temperature. Much of the sensation attributed to sugar is strengthened by molecules that leave the fruit entirely, rising into the air and reaching receptors inside the nose. A ripe strawberry releases some of its chemistry into the air before it is ever tasted. Small molecules escape from the juice and the moist spaces between cells. Heat from the room, warmth from a hand, or the pressure of a knife gives more of them enough energy to leave the liquid surface. Chemists describe these molecules as volatile because they evaporate easily. Once airborne, they can enter the nose and reach millions of smell receptors high inside the nasal cavity. More than 360 volatile compounds have been reported across studies of strawberry fruit and cultivars. A laboratory instrument can detect many substances present in extremely small amounts. Only a much smaller group makes a strong contribution to the scent people recognize. Sensory importance depends partly on concentration. It also depends on the detection threshold, which is the smallest amount a person can smell. A compound with a very low threshold may shape the aroma, even when only a trace is present. Another molecule can be far more abundant while contributing little because the nose responds weakly to it. Several chemical families work together in ripe strawberries. Esters supply many of the bright, fruity notes. Ethyl butanoate can suggest pineapple or other tropical fruit. Methyl butanoate has a sharp, sweet fruitiness and can be detected at low concentrations. Strawberry cells create esters by joining alcohols with organic acids through enzymes active during ripening. The exact mixture changes with cultivar, temperature, and harvest stage. Furannones form another important group. Furanil contributes sweet impressions often compared with caramel, cotton candy, or cooked pineapple. Its relative, mesiferane, carries an added methyl group and produces a warm, sweet aroma with a slightly spicy quality. Some cultivars make large amounts of mesiferane. Others produce very little because they carry a less active form of the enzyme needed for its final chemical step. Terpenes add lighter floral qualities. Linalool is one of the best-known examples in strawberries. It can smell floral with a soft citrus-like edge. The compound also occurs in lavender, basil, citrus peel, and many other fragrant plants, although its effect changes when it is blended with different surrounding molecules. Certain strawberries contain gamma-decalactone, a lactone associated with peach-like aroma. Its presence can make one cultivar seem especially soft and ripe in scent, even when its sugar level resembles that of another berry. Green fruit carries a different balance. Hexanol and cis-3-hexanol contribute leafy, cut grass impressions. These compounds are made from fatty acids when plant tissue is damaged or when enzymes become active during development. Their relative importance often declines as fruity esters and sweet furannones increase. Organic acids also contribute faint odors of their own. Trace sulfur compounds can matter at concentrations so low that ordinary chemical measurements may barely suggest their sensory power. Sulfur molecules often have exceptionally low detection thresholds, allowing tiny quantities to alter the finished aroma. Researchers separate this mixture with gas chromatography. A sample of strawberry vapor is carried through a long, narrow column. Different molecules travel at different speeds because each interacts differently with the material inside the column. They emerge one by one rather than as one blended scent. A mass spectrometer can then break the molecules into charged fragments and help identify them from the pattern. Identification alone does not reveal what the human nose notices most strongly. Gas chromatography olfactometry adds a trained person to the process. Part of the separated sample flows toward an instrument, while another part reaches a small smelling port. The assessor records when a fruity, green, floral, or caramel-like odor appears. This allows researchers to connect a measured compound with a real sensory impression. Strawberry aroma reaches the nose by two routes. Orthonasal smell occurs when molecules enter through the nostrils before the fruit is eaten. Retronasal smell begins during chewing. The teeth crush cells, saliva warms the juice, and volatile compounds travel from the mouth through an internal passage into the nasal cavity. The brain combines those signals with sweetness, acidity, temperature, and texture. Together, they form the single experience called strawberry flavor. Because every cultivar builds this chemical mixture in different proportions, Two berries of equal size and color can taste remarkably different. Two strawberries can reach the same deep red color and still release very different aromas when cut. Their sugar levels may be similar. Their shapes may appear almost identical. Yet one carries a strong, sweet fragrance, while the other seems mild. Part of that difference begins in the genes inherited by each cultivar. Genes provide instructions for building enzymes, the proteins that carry out chemical reactions inside living cells. Different versions of one gene may produce an enzyme that works strongly, weakly, or hardly at all. Those differences can change which aroma molecules a strawberry makes during ripening. One example involves a gene known as FAOMT. It carries instructions for an enzyme that converts furanyl into mesiferane. Furanyl provides sweet notes often compared with caramel or cooked pineapple. Mesiferane adds a warmer, slightly spicy quality. Some cultivars carry a highly functional version of this gene and produce clear amounts of mesiferane. Others make very little, even when their fruit reaches full ripeness. A second gene, called FANES1, is associated with the production of linalool and related terpenes. Linalool gives some strawberries a floral, citrus-like scent. A cultivar with strong activity in this pathway may smell brighter than one whose aroma depends more heavily on fruity esters. Gamma-decalactone shows a similar genetic pattern. This peach-like compound appears strongly in certain cultivars and may be nearly absent from others. Genes establish the chemical possibilities, while growing conditions influence how strongly those possibilities appear. Sunlight affects photosynthesis in the leaves and helps regulate the flavonoid pathways involved in color and related compounds. Fruit developing under dense shade may accumulate pigment and sugar differently from fruit receiving steady light. Temperature changes the speed of nearly every reaction inside the plant. Warm conditions accelerate flowering and ripening. When fruit develops very quickly, it may have fewer days to import sugar and build a full mixture of volatile compounds. Cooler nights can slow respiration and preserve more of the carbon gathered during the day, although prolonged cold also delays growth. Water supply creates another layer of variation. Heavy irrigation or rainfall close to harvest can cause receptacle cells to expand rapidly. The berry grows larger as water enters, which may dilute its soluble sugars. Severe dryness reduces photosynthesis and limits fruit size, so the strongest flavor does not come from simply withholding water. Nutrient supply and growing medium also influence metabolism. A plant rooted in field soil may experience changing moisture and mineral availability. Plants raised in coconut fiber, peat-based substrate, or hydroponic systems receive nutrients through carefully mixed irrigation water. The result depends on how well those conditions match the needs of the cultivar. Crop load matters as well. A plant carrying an extreme number of berries must divide imported sugars among them. With a moderate number of fruit, more assimilate may be available for each one. Assimilate is the general name for sugars and other materials produced through photosynthesis and moved into growing tissue. Harvest timing can outweigh many earlier advantages. A berry picked when only part of its surface is red has had less time to receive sugar from the leaves. Several aroma pathways are still increasing during the final days on the plant. Later storage may soften the tissue and slightly deepen the color, but it cannot replace all of the flavor development lost through early harvest. This helps explain why strawberries bought close to the farm can sometimes taste especially rich, They may be selected for softness and picked at a later stage because they face only a short journey. Commercial berries, sent hundreds or thousands of kilometers, need enough firmness to survive repeated handling and refrigeration. Neither growing system guarantees superior flavor. A local cultivar can be bland when harvested early or grown under poor conditions. A shipping cultivar can taste excellent when fully ripe and carefully handled. The same named cultivar may also perform differently across two regions or between separate years. This is called a cultivar by environment interaction. Its genes remain largely the same, while heat, light, water, crop load, and harvest date change how those genes are expressed. Storage continues altering the fruit after picking. Some volatile compounds decline, while others shift as cells soften and respiration consumes stored sugars. These changes can begin before any visible mold or shriveling appears. Strawberry flavor is therefore a temporary result of inheritance, development, growing conditions, maturity, and handling. The strongest aroma and sweetness usually arrive close to full ripeness. At the same time, the berry's supporting tissues begin losing their strength. As aroma strengthens and sugar accumulates, the same receptacle is becoming easier to damage. A green strawberry resists pressure because its cells are supported by firm walls and held tightly against their neighbors. Water inside each cell also pushes outward against the wall. This internal pressure is called turgor. A well-watered, unripe fruit has high turgor, giving its tissue a crisp, dense feel. Each plant cell is surrounded by a primary cell wall. That wall contains thin strands of cellulose called microfibrils. They are embedded within a moist framework made from pectin, hemicellulose, proteins, and water. Cellulose gives the wall strength. Hemicellulose helps connect and organize its different parts. Pectin fills much of the space between them and plays an important role in holding neighboring cells together. Between adjacent cell walls lies a thin layer called the middle lamella. It is especially rich in pectin and acts like a natural cement between cells. In green fruit, this layer helps the receptacle remain firm even when it is pressed, shaken, or touched by nearby leaves. Ripening gradually changes that structure. Hormonal signals alter the activity of several genes, and the cells begin producing proteins that remodel their walls. No single enzyme causes all strawberry softening. Several processes occur together, and their effects build over time. One enzyme, polygalacturonase, cuts long chains within pectin polymers. A polymer is a large molecule built from many smaller units linked together. As these pectin chains become shorter, the wall framework loses part of its strength. Pectate lyase acts on certain forms of pectin after their chemistry has been changed. It splits those molecules through a different kind of reaction. Pectin methylesterase removes small chemical groups from pectin. This can make parts of the pectin network easier for other enzymes to attack, although the exact result depends on where and when the enzyme acts. Proteins called expansins loosen the connections among wall materials. They do not digest the wall directly. Their action allows cellulose and surrounding compounds to shift slightly in relation to one another. As these changes spread through the receptacle, the middle lamella weakens. Cells begin separating more easily when pressure is applied. The walls themselves also become more flexible. This produces the texture expected from ripe fruit. Teeth pass through the receptacle with little force. Cells break open, releasing juice containing sugars and acids. Volatile aroma compounds escape from the damaged tissue and travel into the nasal cavity during chewing. The qualities that make a strawberry pleasant to eat, therefore, depend partly on controlled structural weakening. Researchers measure this change with firmness tests. A compression test presses a flat surface against the berry and records how much force is required to deform it. A penetration test pushes a narrow probe into the receptacle. Breeders can compare those readings across cultivars, harvest stages, and storage conditions. A ripe shipping cultivar may withstand greater force than a soft variety intended for nearby sale. Handling after harvest adds pressure in many forms. A berry can be squeezed by a picker's fingers, pressed beneath another fruit, or struck against the wall of a container. When the force exceeds what the softened tissue can absorb, cells rupture beneath the surface. This damage creates a bruise even when the skin does not appear broken. Released cell contents provide moisture and nutrients that microbes can use. The damaged area also loses some of its normal chemical defenses. Water loss causes a different kind of decline. After picking, moisture continues evaporating through the fruit's surface and through the opening left near the calyx. As cells lose water, turgor falls. The receptacle becomes less plump, its shine fades, and the surface may begin to wrinkle. High humidity can slow this loss, while very dry air speeds it. Ripeness has therefore brought the strawberry into a narrow period when flavor is strong and physical protection is weak. Once the stem is cut, respiration continues and water keeps leaving the fruit, while the leaves and roots can no longer replace what has been used or lost. The moment a strawberry is detached from its stem, its connection to the plant's water and sugar supply ends. Its cells remain alive. They continue taking in oxygen and breaking down stored sugars through respiration. This process releases usable energy, along with carbon dioxide, water, and a small amount of heat. Strawberries have a relatively high respiration rate compared with many longer-lasting fruits. Their soft tissue, thin outer surface, and high water content leave them with a short post-harvest life. Warmth speeds nearly every part of that decline. Enzymes work faster as temperature rises. Stored sugars are consumed more quickly. Cell walls continue weakening, and water escapes more rapidly into the surrounding air. Fungi and bacteria also grow faster when conditions become warm and moist. A container of strawberries left in summer field heat can therefore lose useful shelf life within a few hours. Cooling must begin soon after harvest. Simply carrying warm berries into a cold room works slowly, because chilled air does not move evenly through tightly stacked cartons. The fruit near the outside cools first, while berries near the center may remain warm. Commercial operations often use forced air cooling. Cartons or pallets are arranged beside a fan, and a cover directs cold air through the vents in the packaging. The fan creates a pressure difference that pulls chilled air around the individual berries. This removes field heat much faster than leaving the containers in still air. Clear plastic clamshells are designed with slots and holes partly for this reason. The vents allow cold air to pass through during cooling and later permit some heat and moisture to escape. Poorly aligned vents or tightly blocked cartons slow airflow and leave warm pockets inside a load. Recommended storage is close to 0 degrees Celsius or 32 degrees Fahrenheit. The fruit itself should remain just above its freezing point. since ice crystals can rupture cells and leave the tissue watery after thawing. Relative humidity is usually kept near 90 to 95%. Relative humidity describes how much water vapor the air contains compared with the maximum it could hold at that temperature. High humidity slows evaporation from the fruit and helps preserve its smooth, full surface. Air that becomes completely saturated can create condensation. Free water collecting on the berries provides favorable conditions for fungal spores to germinate. Storage, therefore, requires a careful balance between limiting water loss and keeping surfaces dry. Some shipments use a modified atmosphere with carbon dioxide levels around 10 to 15%. Carbon dioxide at these concentrations can slow respiration and suppress the growth of certain decay organisms. The treatment must be controlled. Excessive carbon dioxide may injure the fruit, alter its color, or produce unpleasant flavors. The berry's journey depends on an unbroken cold chain. After forced air cooling at the farm, cartons enter a refrigerated truck. They may pass through a distribution center before reaching a store's chilled display. The final stage continues inside a household refrigerator. Every warm interruption consumes part of the remaining storage life. Cooling the fruit again slows further change, though it cannot reverse softening, water loss, or sugar already used during the warmer period. Handling at home can shorten the same narrow window. Washing strawberries before storage adds surface moisture and may spread spores from one berry to another. Running water and repeated movement can also bruise ripe tissue. Household guidance usually favors keeping the fruit dry and washing it shortly before eating. Any washed berries stored for later need thorough drying and refrigeration. Cold preserves a strawberry by slowing enzyme activity, respiration, moisture movement, and microbial growth. Those processes never stop completely. Even under ideal conditions, the fruit continues moving away from the brief state it held at harvest. Refrigeration can delay visible decay, while spores already present on flowers or fruit surfaces remain ready to grow when moisture and temperature become favorable. A strawberry flower can be exposed to fungal spores before its petals have fully opened. One of the most important species is Botrytis cinerea, the fungus that causes grey mold. Its spores move through the air, ride on splashing water, and spread by contact between infected and healthy tissue. A spore landing on a petal, stamen, or damaged part of the flower may begin growing when moisture remains on the surface long enough. Aging flower parts provide an especially useful starting point, as petals and stamens senesce, meaning they enter their natural stage of decline. Their cells weaken and release nutrients. The fungus can grow through this fading tissue and reach the young receptacle nearby. Visible decay may still be absent. This quiet stage is called a latent infection. Latent means present without obvious symptoms. The fungus may remain limited while the strawberry is green, firm, and chemically resistant. Ripening changes those conditions. Cell walls loosen, sugars become more available, acidity shifts, and some defensive systems weaken. The same changes that improve flavor and texture make the receptacle easier for the fungus to enter and use. After harvest, a hidden infection can develop rapidly. A bruise breaks cells and releases juice. Condensation adds surface water. Warm air speeds fungal growth, while berries touching inside a container create short paths from one fruit to another. Grey mold often begins as a soft, pale area that later develops the dusty grey spores responsible for the disease's name. Cool to moderate temperatures can still support this fungus when humidity is high, which is why refrigeration slows grey mold without eliminating it. Disease can also begin far below the fruit. Macrophomina phaseolina is a soil-borne fungus that causes charcoal rot in strawberries and many other crops. It attacks roots and crown tissue, interfering with the movement of water through the plant. Leaves may wilt during hot weather because damaged roots cannot replace water as quickly as it is lost. The disease is often most severe when high temperatures occur together with dry soil and plant stress. Macrophomina survives between crops through microsclerotia. These are tiny, dark, hardened structures made from tightly packed fungal cells. Microsclerotia can remain in soil or infected plant debris until roots from a suitable host grow nearby. Their durability makes the pathogen difficult to remove once a field becomes heavily infested. Breeding offers one way to reduce the damage. In one recurrent selection program, researchers began with a population in which only about 1% of plants were considered highly resistant to charcoal rot. Recurrent selection means choosing the strongest individuals, crossing them, and repeating the process with their descendants. After 2 selection cycles, the share of highly resistant plants rose to about 74%. That result shows how quickly useful alleles can become common when resistance is measured carefully and selected across generations. Resistance usually remains quantitative. Several genes each contribute part of the protection, producing a continuous range from highly susceptible plants to strongly resistant ones. Field conditions still influence the result. A resistant cultivar may suffer when heat, drought, and pathogen pressure become extreme. Growers therefore combine genetics with clean nursery plants, crop rotation, good drainage, sanitation, and irrigation that avoids prolonged wetness around flowers and fruit. Open foliage improves air movement and allows surfaces to dry more quickly. Damaged or infected berries are removed, so they do not become sources of spores. Fungicides may also be used at carefully chosen times. Repeated use of one mode of action can select fungal strains able to survive it. Rotating products with different modes of action helps slow that process. Bloom treatments require particular care because pollinating insects are active in the flowers. Application timing must also follow residue limits that regulate how much pesticide may remain on harvested fruit. Disease develops through the combined effects of pathogen, host, weather, and management. The strawberry's shallow roots, delicate flowers, soft receptacle, and brief storage life place several forms of risk into one crop. Modern fields are built around those limits, using beds, mulch, irrigation lines, protective covers, repeated harvests, and rapid cooling to keep the fruit within its narrow window of quality. A modern strawberry field is shaped to control water before the first crown is planted. Growers form long raised beds above the surrounding soil. These beds improve drainage by giving excess water a path downward and away from the crown. They also create a loose root zone where shallow feeder roots can find oxygen. Keeping the crown above saturated ground reduces stress and lowers the risk from several root and crown diseases. The extra height makes harvesting easier because the fruit sits farther from the lowest part of the field. A sheet of plastic mulch is often stretched tightly across each bed. black plastic absorbs sunlight and warms the soil beneath it, which can encourage faster root growth during cool parts of the season. It also blocks light from reaching many weed seedlings. Fewer weeds leave more water, nutrients, and space for the strawberry plants. The mulch separates developing berries from damp soil, reducing mud, surface damage, and some routes of infection. Other mulch colors change the field in different ways. White or reflective surfaces remain cooler under strong sunlight. Certain films reflect light upward into the foliage and around fruit hidden beneath leaves. A narrow drip line usually runs beneath the plastic. Small openings release water close to the root zone instead of wetting the entire bed. Dissolved fertilizer can be added to this water through a method called fertigation. The system allows growers to adjust nitrogen, potassium, and other nutrients as the crop moves from leaf growth into flowering and fruit production. Drip irrigation also leaves most flowers and fruit dry. That can reduce disease pressure compared with overhead sprinklers, which spread water across leaves and may splash spores from one plant to another. Protective covers add another layer of environmental control. A low tunnel is a small plastic or fabric cover, supported by hoops directly over a row. It can protect flowers from light frost, reduce wind, and keep rain from striking ripe fruit. A high tunnel is large enough for workers to enter. These walk-in structures can extend the season by warming the air and soil during cool weather. They also reduce direct rainfall, which helps keep fruit surfaces dry. Ventilation remains important because trapped heat can rise quickly on a sunny day. Pollinating insects also need access during flowering, so sidewalls or tunnel ends may be opened when conditions allow. Different production systems arrange the plants in different ways. Annual plasticulture places crowns through holes in mulched raised beds and often keeps them for one main crop cycle. Matted row systems allow runners to root and fill a permanent strip, creating many daughter crowns for the following season. Day-neutral strawberries may grow in raised beds, containers, tunnels, or elevated tabletop gutters that bring the fruit closer to a worker's hands. The harvest itself cannot happen in one pass. Flowers open at different times, and berries on the same plant reach full color on different days. During peak production, crews may return every 2 or 3 days. Workers search beneath leaves, judge color and firmness, and detach each ripe berry near the calyx. Whenever possible, the fruit goes directly into the same clamshell that will reach the store. Fewer transfers mean fewer chances for bruising. Labor becomes one of the largest costs in strawberry production because the crop demands slow, careful decisions. A red berry may be hidden behind foliage. Another may be partly ripe, with a pale shoulder beneath the green cap. Soft fruit must be placed gently enough to avoid pressure damage. Robotic harvesters are being developed, though strawberries remain difficult targets. Computer vision must locate fruit partly covered by leaves, judge ripeness under changing light, and separate one berry from several nearby fruits. A gripping tool must reach the stem or calyx without squeezing the receptacle. The machine must also avoid damaging green berries that still need several more days on the plant. After picking, the field system connects immediately with the cold chain. Clamshells move into cartons designed for airflow. Cooling equipment removes field heat. Refrigerated trucks carry the fruit toward distribution centers and retail displays. Harvest schedules, packing crews, coolers, and transport all have to align with the berry, whose best condition may last only a few days. Centuries of farming have therefore reshaped the environment around the strawberry. Current research is beginning to reshape the plant and its surroundings together through genome mapping, environmental sensors, crop models, and machines able to watch each berry as it develops. That closer watch begins inside the strawberry's 56 chromosomes. Reference genomes map DNA across 8 related chromosome sets. Small differences in those maps can serve as genetic markers, allowing researchers to follow regions linked with flowering time, firmness, disease resistance, color, sugar concentration, and aroma. The task remains unusually complex because several related copies of a gene may influence the same trait. One copy can be highly active in the receptacle, while another works mainly in roots or flowers. A breeder studying firmness may therefore need to track several regions at once. Genomic selection expands that approach across the entire genome. Researchers first measure large groups of plants in fields or greenhouses. They record yield, fruit quality, disease response, flowering dates, and other traits. Then compare those results with thousands of genetic markers. A computer model learns which marker patterns tend to appear in the strongest plants. New seedlings can receive a predicted breeding value before every candidate completes years of field trials. Prediction helps breeders decide which plants deserve further field testing. The useful combination is changing as growing conditions shift. Hot flowering periods can reduce pollen fertility. Dry soil places greater strain on shallow roots, while intense rain increases fruit wetness and disease pressure. Irregular winters may disturb dormancy and flower development. Pathogens can also expand into regions where colder conditions once limited them. Breeding programs increasingly seek flowers that remain fertile during heat, roots that keep working under stress, and resistance that lasts across several pathogen strains. Firm fruit still needs a pleasant texture, while improved shelf life must preserve the sugars and volatile compounds that give the berry its flavor. Some growers move part of this work into controlled environments. Plants may be rooted in peat-based mixtures, coconut coir, rock wool, or other manufactured media. Water and dissolved nutrients arrive through fertigation lines, sometimes in systems that collect and reuse part of the drainage. Light-emitting diodes allow growers to adjust light intensity, daily timing, and the balance of wavelengths reaching the leaves. Sensors can measure temperature and humidity around the plants. Other instruments track carbon dioxide, root zone moisture, light, and electrical conductivity, which estimates the dissolved salts in the nutrient solution. Crop models combine these measurements with known rates of flower and fruit development. They can estimate when blossoms will open and when a wave of berries may reach harvest. In one climate chamber study, researchers adjusted temperature to shift predicted strawberry harvest peaks. The intended harvest date was reached within approximately 1 day in 3 of 4 treatments. This forecasting could help farms coordinate labor, cooling space, and deliveries. Robotic harvesting adds another layer of prediction. Cameras identify fruit among overlapping leaves. Depth sensors estimate position, while software classifies ripeness and plans a path through the plant. A gentle end effector, the tool at the end of the robotic arm, must detach the berry without pressing into the softened receptacle or striking nearby green fruit. Controlled systems carry their own costs. Buildings, pumps, lamps, cooling equipment, and sensors require money, electricity, maintenance, and manufactured materials. Field farming will remain useful wherever climate, land, and labor make it practical. Protected systems offer another way to produce fruit in difficult seasons or close to large markets. The strawberry held at the beginning can now be seen in full. Its green calyx once enclosed a flower bud. The red flesh is an enlarged receptacle carrying hundreds of achenes, each surrounding one seed. Signals from those developing fruits help shape the tissue beneath them. A compact crown divided water and sugar among roots, leaves, runners, stored reserves, and fruit. 8 chromosome sets preserve ancient hybrid events, while 2 American species met in European gardens only about 3 centuries ago. Breeding enlarged the receptacle and strengthened it for travel. A BA helped guide the final ripening stage. Anthocyanins supplied the red color, and volatile molecules gave the fruit its recognizable scent. Cell wall remodeling made the flesh soft enough to release its juice. Cooling slowed the decline that began as soon as the stem was cut. For a brief interval, hundreds of separate fruits rest across one red receptacle, while color, aroma, sweetness, and softness arrive together.